Everybody, thank you for attending this Lychee Erinose MIte webinar update. I'm Jeff Washulewski, Commercial Fruit Crops Extension Agent. We're also going to have Daniel Carrillo, Entomologist in Tropical Fruit, talk about the research update on the biology and control of the Lychee Erinose MIte. And Dr. Jonathan Crane, Tropical Fruit Crop Specialist, will be talking about the current LEM mitigation and control recommendations. So that's our order there. I'll go first with an introduction. At the end, we're going to have a survey for commercial growers. So I'll put that link into the chat. Then we'll do questions and answers. So hold those to the end. You'll be able to do those in the chat because you are on mute and your video is off. So your microphones and cameras will be off. Hold your questions until the end. This is being recorded, so it should be on YouTube by the end of the week. So if you need to look at some of the slides, they'll be there. You'll be able to ask questions at the end through the chat. I'll be looking at that. Like I said, we'll do a quick survey before the Q&A using the link that I'm going to provide to you. We also have some handouts, so I'll be putting those in the chat as well. There's 4 of those. Some of them are, how to control or work with Lychee Erinose MIte. So we're putting those there. So the Lychee Erinose MIte in Florida. Let's talk about the plant itself. It's in the Sapondaceae imported fruit crops within the family, longan, rambutan, pulsan, and ackee. We can grow longan and ackee here. LEM, luckily, only attacks lychee and not the others. History in Florida: in 1955, it came into Sarasota County. 1993 in Coral Gables. In Miami-Dade County, it was eradicated both times. However, in February of 2018, it was found in a 3-acre commercial lychee grove in Lee County on Pine Island, and it was not able to be eradicated. As you guys know, you're dealing with it. It's moving around. I see someone has raised their hand, so let's keep the questions until the end. LEM general information, like I said, it only attacks lychee, not longan. At the very beginning, we thought it might. It really likes new leaf fleshes, and they're the most susceptible. It forms these small blisters, as you can see in the picture there, and silver white colored hairs that are under the leaf. You can kind of see some of them there. This progresses to a reddish brown hairy mass. It damages the fruit, which is a big problem. The flowers, another big problem. Stems and leaves. And then here's what it looks like when it's been there for quite a long time. You have that reddish brown hairy mass. The mites are too small to see with the naked eye. You look at the arrow, there's one in a big, big, strong microscope. So they're so small, they can move by air. They can move on tools, clothes, and even on honeybees. So what's the importance? Florida is the leading producer of lychee in the country. Nearly 70% of that is in Miami-Dade County, and the pest can result in an 80% reduction in fruit production. So you see that's a big problem. For more information, we're going to put the latest stuff into the chat, so look for it there. You can also search Miami-Dade Extension or Trek Lychee Erinose MIte. So Miami-Dade Extension Tropical Fruit or Trek Lychee Erinose MIte, you'll find information there. Miami-Dade Extension Tropical Fruit is not updated yet, but it will be by the end of the week. So you get fact sheets there and the label for what you need to spray. If you are a commercial grower or an extension agent, you can reach me at my email there or my phone number, direct line 305-679-0227. Homeowners, you're going to go to Veronica Charpentier, and her number is 305-679-0228. Thank you for your time. We're going to move on to Dr. Carrillo. Remember, hold your questions until the end, and we are recording, and you can ask questions at the end through the chat, and then we'll have the survey. So here's our next speaker, Dr. Carrillo. I'm going to stop sharing, and you should be able to go Go ahead, Doctor.
Thank you, Jeff. Give me 1 second. Second. Can you see the presentation there?
Yes, sir.
Okay. Well, thank you, Jeff, and thank you everyone for joining. I'm going to try to summarize almost 8 years of dealing with this mite since we found it in February 2018. And I have a lot of authors here are probably missing many, but virtually everyone that has worked for Dr. Revinti's lab or my lab in the past few years has worked and helped in the research dealing with this mite. I want to acknowledge especially Livia Ataide and Jacqueline Francosi de la Vecchia, two postdocs that really took the lead and contributed a lot to the research. But we also have international collaborators, a group from Brazil that is working with the surfactants. We will see more about this later. So this is the little guy, tiny guy that we're dealing with. Scientific name is Aseria lychee, or the Lychee Erinose MIte. It is among the smallest arthropods that can infest a plant. This particular family of mites are very tiny, so we are basically dealing with something that we cannot see. We mostly see the symptoms, the damage, what they cause on the plant. They have their mouth parts are stylids that are sort of some needles that they use to pierce and feed on the leaf epidermal cells. And the problem is not so much with this feeding or the piercing that you can see here in this scanning electron micrograph. These holes that you see here, these are the feeding sites. But the mite injects an unknown substance that induces the proliferation of these hairy masses. We call them trichomes. We don't know exactly if they are trichomes or not. But yeah, the issue is that the mite is inducing these malformations on the leaves. Like Jeff said, they are tiny. You need magnification, high magnification to see them. This is how you would see them in the lab under a scope. But for the most part, they are embedded there inside these hairy masses protected from the environment. More pictures here. You see a bunch of mites here on the left and the eggs here at the base of those hairs or the trichomes on your right. So part of my responsibilities as an entomologist in tropical fruit crops is to know what the problems of the different crops are. So I had seen this problem before it arrived to Florida. I saw it in Brazil, I saw it in Hawaii. And the damage is so characteristic that when we found it, I received a call by Armando Monterroso that worked for Brooks for many years. He was on Pine Island in Lee County. He described to me, look, my lychees have a very strange hairy mass on the leaves. And only with that, I was 100% sure that we were dealing with the Lychee Erinose MIte. The symptoms are so characteristic. I didn't ask for a picture or anything. Next day, we were driving there. And since then, we have been working with this mite and doing several things. The mite is native to Southeast Asia, but it has been spreading quite a bit. It's also in Australia, in Hawaii, it has been reported for a long time. In Brazil, has caused a lot of damage, was reported in 2010 and has caused a reduction in yield, very significant. More recently, I think last year or early this year was reported in Italy. So one of the first things that we did is try to compare different populations. We worked with collaborators from Australia, Hawaii, India, Brazil, Taiwan, collect populations to see if we had some differences in the populations. And it seems like our population matches exactly the populations from Hawaii, at least in terms of genetic sequences, some genetic sequences. So there are different lineages of this mite. We don't know if one is more aggressive than other, but ours seems to have the same origin as Taiwan. At the beginning, there was some confusion. There are some old publications that suggest that this mite also fits on longan, but now it's absolutely clear that this mite is a specialist. It only feeds and develops on lychee. And this is a characteristic of the family of these mites. No, they tend to be highly specific. So it can only live and reproduce on lychee. The symptoms, Jeff mentioned this. They have these stylets that they have. The mouth parts are very delicate, and they can only penetrate on the very soft tissue. So you usually find the initial symptoms on the young leaves. You can also see blisters. There's a diversity in the symptoms that you can observe. This blister that you see here, a more pronounced leaf curling, like in this slide. Then this is the erinium, what I was talking about at the beginning. Usually starts on the underside of the leaves. These hairy masses, we're going to talk a lot more about this erinium. But this can grow, and it can affect the entire leaves and also grow on the stems. I want to point out is not that this didn't start, the damage didn't start when the stem was already formed. The damage started or was caused when you have buds, when you have the tissue was very soft and it was still growing, and then it expresses like this, right? You can also see damage on the panicles, flowers, and on the fruit. But again, the mite does not cause this on a fruit that is already formed. It will infest the fruit when it's small. As the fruit develops, you will observe the erinium. So this erinium also, as the mite, it has a cycle, and has some different stages. One first stage we sees for the color of the erinium, hyaline, when those hairs are almost transparent and small little spots on the leaf. Then it turns almost white. Then it becomes amber, like it has this reddish color, and finally it becomes dark, almost black. And in sampling all these stages, we noticed that the amber color, that third stage, is when you really have a very large population, when the mite is booming. The hyaline and the white give you an indication that the infestation is starting, but the amber is when you have a really prolific multiplication of the mites. And the dark brown is when the erinia dies off. We also studied the effects of the temperature, and this is quite important. The mites really, and the erinium, they like moderate temperatures. For example, the erinium really develops this amber color develops more when you have cooler temperatures, 15 degrees Celsius or 60 degrees Fahrenheit. And the mite itself has a maximum reproduction at around 70 degrees Fahrenheit or less 22 Celsius, right? So that's another reason for making this workshop right now. We're going to enter the period where it's optimum environmental conditions for these mites. So in general, the environmental conditions that are favorable for this mite are periods where you have new growth and you have moderate warm and dry periods. When you have high temperatures, high relative humidity, and high rainfall, we see a drop in the populations. I told you that I had seen this problem in Hawaii before, and over there, you have a very clear example of how the environmental conditions affect this mite. On the Big Island, you have two distinct areas. If you go to the west part of the island, in Kona, it's very dry and they have very low precipitation, sometimes less than 30 inches per year. Over there, it's a very insidious pest and almost impossible to grow lychees in this area. Whereas if you go to Hilo, that is to the east, where it's very humid with more than 100 inches of rain per year, but sometimes 300 inches per rain. Over there, the mite is a lot milder. It's not so problematic. So it's really, really important, the effect of these environmental conditions, the humidity and the rainfall on this mite. Now in Florida, we had the opportunity to really follow the expansion and the colonization patterns of this mite. So here at the station, we have a small grove with 190 trees approximately, 3 varieties, Mauritius, Sweetheart, and Brewster. And we managed to detect the first tree when we got the first infestation, well, the first tree that was colonized by the mite. And we did biweekly evaluations involving almost everybody in the labs because we had to inspect everything with a lot of detail to see where the infestations were progressing. So I'm going to show you these heat maps that show how the infestation progressed. The black spots is because the trees are missing, but the intensity of the green color shows the intensity also of the infestation by the mite. So this starts 90 days after the first detection. The first 90 days were relatively slow, but it's 150, 120 days, 135, 150, 165, 180 days, 195, 210, 225, 240, 255, 270, 285, and 300. So basically, we had a period of slow expansion the first 90 days, and then in approximately 100 days, 80% of the trees got infested. And in less than 1 year, 100% of the trees, all the trees were infested. So it really has an incredible ability to move. In less than 1 year, all the trees had the mite, and most likely every lychee tree in Florida is likely to be infested. It will be infested. I've seen this also in Brazil, in Hawaii, where you have this mite. It becomes part of the system. So this is something very conceptually very important that you have to understand, that the mite is not going anywhere, and it will likely be infesting your trees. So that is very important for the management. We are not going to get rid of this mite. We just need to learn how to deal with it. So how it disperses? First, by drifting on air currents. The number of mites in this Erinium is incredible. Just to give you an idea, if you take a small section with a punch hole like this, you can get more than 2,000 miles in this little circle. So the number of mites that are infesting a tree are in the hundreds of thousands, maybe millions. So once you have they get the airborne, they are blown in many directions, and out of chance they fall in the other trees. They obviously cannot direct their flight, but it's just the sheer number of mites that makes this high probability of getting infested. They are also moved by honeybees. There's some indication that the mite becomes more active during the flowering period. There's some documents from Australia that suggest this. We tried to study this and did a very detailed study on the volatiles that are released by the different plant parts, and we didn't observe this. But it does seem like the mites have some sort of association with the flowers. And anyone that has a lychee tree, you know that the flowers are very attractive to the honeybees. So this period, the flowering period, is a period where you could expect a high dispersal of the mite. And of course, through plant propagation, lychees are propagated through air layers. So if you propagate a tree that is infested, most likely you are moving the mite. And this is how it arrived to Florida. This is the only way that the mite could have arrived to Florida, only on a live plant. No, the mite cannot live anywhere else. It needs a live tissue to be transported, or at least survive for prolonged periods. In our trial here, we also observed that the Mauritius seems to be more susceptible, followed by Sweetheart, and less Brewster. However, this needs to be studied in more detail, because it was not we don't have the same number of trees, but there's some indication that there are differences among the varieties in terms of their susceptibility to the mite. So now that's more or less what we know about the biology. So it's a very persistent mite that is here to stay. All trees have the mite, and what we need to understand is that we want to see when it is worth to protect the tree. We're going to see how we build this strategy of how to control this mite, and Jonathan is going to talk about this later as well. But we use what is called integrated pest management. It's based on the pest ID. In this case, the symptoms are so unique that you don't really need to submit many samples or do a lot to know that you're dealing with the Lychee Erinose MIte. You should monitor, but you should not really monitor for the mite. You should monitor for the conditions that are favorable for the mite. You already know that you have the mite. So what you really need to be aware is that the symptoms and the tree flushing patterns, remember, these stages of the erinium. You know that when you have this amber color erinia, you have a high mite population and high spread risk. Also, when you have new growth, this is what the mite likes. So this is a critical period for protection. And also, when you have moderately warm periods, more or less 70 degrees Fahrenheit, this is when you have more problems with this mite. So you are more monitoring for these conditions. If you want to make an analogy, let's say that you have you're allergic to something, and there are some conditions that trigger outbreaks of this allergy. This is more or less what is happening. So I know that if I am allergic to dust, if I go into a place that is dusty and has a lot of humidity, I'm gonna have an outbreak of the symptoms. This is more or less how we should explore the management of this mite. So when you have the conditions, you really need to be ready to act. You know that you have them, but in other periods, maybe it's not necessary to do anything. So in chemical control, we've made a lot of studies evaluating different acaricides, different miticides of different groups. Sulfur is important to note. It is a very old acaricide, very effective. It can be used in organic production, and it's even used sometimes as a nutritional treatment. So this was the first miticide that we worked with, and the one that we have used more successfully, and we used them always as a reference. So we had a different approach with this mite. Since we cannot see it, it's difficult to count. We made some evaluations using the symptoms of the plant. So we established some bioassays. Since we have a healthy plant, you apply a treatment and then release mites to see if the treatment protected the plant. This we call a prophylactic or a preventative treatment. So we basically have a plant, apply the treatment, then infest it, and then look at the new growth to see if the mite established or it didn't establish. And the other type of treatment is you have a plant that is already infested, and then apply the treatment and then look at the new growth and see if you control the population or not, or if it continues to develop. So we did a series of greenhouse and also field trials. In the greenhouse trials, we found that sulfur and also these three products, fenpyroximate, pirivadem, and tolpenfirat, completely protected the plants. So you have a new plant that is healthy, and you apply these treatments, you prevent the mite from establishing on those plants. All the other acaricides had little effect, at least comparable to water, not different to a water treatment. Interesting that these three acaricides, the three miticides, are of the same group. This mode of action group is 21A. So there seems to be something with this group that seems to be effective with these mites. Now, with the curative treatments, if you have a tree that is infested, has the mite, and you apply the same treatments, no effect. So you are not controlling the mite that is already there. What you can do is to protect a new tissue from the mite. So that is critical again for the strategy of control. The mite is there; it's going to continue being there. What we're going to try to do is to protect the new growth of the plant. Now, in the field trials, they did a similar evaluation strategy. So we went and we tagged new flushes, new buds, then applied treatments, and they followed the development of those new flushes to see if they get infested with the mite or not. In the first field trial, we tested fenpyroximate versus sulfur. And as you can see here, the treatment protected almost completely the new growth from the mite infestation compared to the control. But it was necessary to make 4 applications. In a second trial with pirivadem, we have similar results, but only with 2 applications we managed to have complete control, complete protection of the new growth with pirivadem and sulfur, and no difference between pirivadem, sulfur, or fenpyroximate and sulfur. So very positive results. We have, again, good prophylactic treatments. We have good treatments to protect some critical periods in the growth of the plant, like the panicle emergence and when you have a new flush. So that's our strategy, is to protect these critical periods. Jonathan is going to explain this in more detail. But now we are limited mostly to sulfur. It's the only one that is really registered for use. It's a good acaricide, but also has some limitations. You you cannot apply it at very high temperatures because it can cause phytotoxicity. More than 90 degrees, you're in risk of damaging the foliage. And it is very highly incompatible with other pesticides, with oil, so it's difficult to mix with other products. It's also very easily washed off. So we have these other treatments, fenpiroximate, pirivadem, and tonfelpirat. Tonfelpirat we haven't tried in the field, but fenpiroximate and pirivadem with very good results, as effective as sulfur. But they have other attributes. For example, fenpiroximate is regarded as reasonably safe to honeybees. You have to take this with a little bit of caution, but it's much more compatible than the other acaricides with the honeybees. It's important because the flowering period, as you see, is very important to control this mite. It's also compatible with other pesticides, and especially with oils. But we need registration. We already submit all this data to IR4. We're in the process of getting the labels for this product, but that's also up to the grower associations and the growers to push for this. We can produce the data and continue working on this, but the registration is something that is more responsibility of the agricultural community involving the company that produces the product. So yeah, we have some good protectants, but we don't have curative treatments. And in this, we have a very nice collaboration with the Unesp in Jaboticabal in Brazil, with the Acarolab, with a student that is looking at different adjuvants, surfactants to mix with the acaricides so they can penetrate the rhinium. This picture that you see here, this is more or less the problem why we think we don't have a good control of the mite, because these hairy masses become almost hydrophobic. The water doesn't penetrate, the sprayed solution doesn't penetrate the rhinium. So I don't want to anticipate too much because the student is submitting this for publication, but several adjuvants really enhance the efficacy of the acaricides, especially when combined with pirivadem. So this is another important line of research that we need to continue. We have also tested a number of essential oils to be used in organic production, sustainable agriculture. All the ones that are commercially available, following the same logic with the greenhouse experiments. And we found that among those, Time Guard seems to provide good results, as effective as sulfur. We need to do more field trials to validate this, but preliminary results show some very promising efficacy of this Time Guard. So now moving along to cultural control, pruning. You see here in the grove in our trials, we evaluated pruning. So we pruned with a hedger half of the trees in the grove, half of the rows. And also during the evaluations, when we were monitoring the colonization of the mite in the grove, we observed that the mite usually is in the outer side of the canopy, either in the top, middle, or lower part of the canopy. It's primarily infesting the outer side of the canopy. So it's where the infestation starts, but it's also where you have mostly the largest population of the mite. So you can, by pruning, you can really help in the control, but the pruning has to be followed by a treatment application, a miticide application. If you prune and don't apply a treatment, you are giving the mite substrate to grow. So it has a good effect, the sanitation or the pruning, if it's combined with a acaricide, a miticide application. So you see, in every case, except with abamectin and water, pruning had a good, a beneficial effect in reducing the mite populations. So again, in combination with an acaricide, this is effective by itself. Is not a good idea. Better not do it if you're not ready to apply an acaricide. So pruning has to be directed to the outermost part of the canopy. Pruning has other advantages. Maybe Jonathan is going to talk about that. One of them is that you can synchronize the trees. If you're trying to protect the new flush, you prune them, then you're going to have a period where all the trees are going to be flushing at the same time. So you can apply the treatments and protect it, not different trees flushing at different times, right? And yield, from our experiments, we are not observing that it has an effect on yield, but our trees are very irregular, so I don't want to put the solid data here. Something that we need to explore more. How does pruning affect yield? It obviously has some effect in yield production, but it's things that we need to balance. Once, if you have a very large infestation and you want to reduce it, then pruning might be a good idea. In biological control, we have also worked quite a bit on this and observed a number of predators that naturally visit the Erinium. Amblyssero largoensis here in Florida has also been observed in China. There's a recent publication on this. In general, the mites, these predators, they do feed on the Lychee Erinose mite and they go to the Erinium, but it's not a good food source for them. The predators cannot complete the cycle just feeding on this Lychee Erinose mite. So it's a secondary food for them. So they might play a role in regulating populations, but it's not so critical. We haven't seen it identified a critical predator of this mite. The one that, in my opinion, has more potential is this Phytoceus woodbury. It's reported from Florida and Brazil. It's a smaller predator that has the ability to enter the Erinium and navigate those hairy masses and really find the eggs and the mite inside the Erinium. But we know very little about it. This is a good new project for a student or something. So this mite in particular has that morphology and that shape that can really enter the Erinium. All the others, they just fit from outside. And yes, as you see here in these videos, they do fit. But in terms of regulating the populations, it's unclear what's their role, what role they have. I told you before that it's humid periods and periods with a lot of rain are reduce the populations of the mite. We see a reduction in the populations, and it is associated with the presence of these fungi that attack the mites. We identified one strain of Hirsutella thomsoni, but there are probably several fungi attacking the mites. In general, these fungi are very important regulating populations of this mite and other mites. Naturally, we really don't perceive it, but are very important. Unfortunately, we cannot use them. We don't have commercially available strains of these fungi, but they do play an important role in regulating populations, as you can see here. So this is what we observe in the summer months. The Erinium collapses and a lot of these fungi infesting the mites. Another type of control is through post-harvest treatment. So this is an invasive mite here in Florida and many different parts of the world. And one way to overcome the problem is to sell fruit when it's regulated is to use a post-harvest treatment. So this is one of the first things we did, evaluating dips in paraffinic oil, 3% and 5% solutions of an oil that is commercialized here as stylet, but it's a paraffinic oil. You dip the fruit for 1 minute under constant agitation, and we found that well, I didn't have the image there, but complete disinfestation of the fruit just with 1% solution of dipping 1% solution of this oil, and the fruit quality is preserved. So this is an option that you could use if you are sending the fruit to other areas, other states, other countries, and this is a relatively easy treatment to implement. Also, you don't need special equipment or anything to implement it. Just to summarize, so the Lychee Erinose MIte is a very persistent pest. So it's going to be here. Almost every tree is going to have the mite. We know that moderately warm, dry periods and periods with new growth and periods with amber color Erinia is when you have really high mite populations, high risk of spread and risk of infesting the fruit and really damaging the trees. We have some good prophylactic miticide treatments, and for now we use sulfur, but hopefully we'll have more in the future. And they are highly effective and recommended during 2 critical periods, during panicle emergence and fruit set, and also after topping or hedging the trees to protect the new flush. So Jonathan is going to explain this in more detail, but this is the strategy that, based on the knowledge of the biology of this mite, I think this is what is working best. And moving forward, I think this is a very good strategy to follow. There are natural enemies and abiotic factors that may regulate the populations. We need to understand this a little bit more. And we also need curative treatments, continue this work with the surfactants to really try to kill all the mites that are infesting a tree, if possible. Here, when we started working with this, I called some colleagues in Brazil, and they told me, look, you're going to work 6 months with this mite, you're going to publish 1 paper, and you're going to walk away because it's so difficult. And it's indeed a very difficult mite because you don't see it, and there's the rhinium, quite complicated, but I'm happy to report that we already have 6 publications. We have 3 more on the way. So of course, the work of many people in this. So I think here in Florida, we have produced more information about this mite than anywhere else in the world. Future research, I hope Jonathan is going to talk a little bit about this, nutrition and irrigation in relation to new growth, because these are anything that you do that promotes flushing or new growth on the plant is critical to the management of this mite, right? So you have to be very, very careful not to induce new growth when you don't want it or when you are not ready to protect it. Again, the work with the surfactants and work on symbiotic association. There's a big question about the possible involvement of an algae. This red coloration of the Erinia is associated with an algae that we haven't been able to really understand. So this is something that may be for the future. I want to acknowledge especially the Florida Department of Agriculture and Consumer Services for a block grant that has sponsored a lot of this research, and primarily the control, all the chemical control treatment. Also, USDA ARS, the subtropical station here in Miami, that have a cooperative agreement that has supported a lot of this research. And yeah, virtually everybody that has passed through the labs that had really had to count a lot of mites and spend some times in the field looking at lychee trees. And thank you. I'm going to pass it to Jonathan.
Thank you, Daniel. I want to let everybody know that I put the fact sheets into the chat. There's 4 of them there, so you can get them out if you'd like. I'll put them in there one more time about midway through Jonathan's talk. He's going to talk about current LEM mitigation and control recommendations. Take it away, sir.
All right. So you can see the screen?
Yes.
Thank you, Jeff, and thank you, Daniel. That was a really excellent presentation. Really excellent. Okay, so I'm going to talk a little bit about an update on the Lychee Erinose recommendations for mitigation control. And I want to thank the other people listed on this slideshow. So we've seen that, sulfur is a very good miticide and works very well, as Daniel has shown. We have seen, though, that applying it monthly or every time the tree flushes, and we've seen this in a number of lychee groves, can lead to some phytotoxicity on the leaves. And this includes, yellowing and leaf tip and leaf marginal necrosis, which causes another problem in the tree. So the tree becomes unhealthy. It's difficult for the tree to function. It's also costly and time consuming. If you're doing this, we have seen a number of growers applying it, at least monthly and maybe more often. And I totally understand why, because you want to keep the mite population from colonizing any growth on the tree. But we've also seen that applying sulfur, all the time, you still may get some lem infestation in the sense that you're not going to cover 12 months of the year. Usually you'll miss a flush or parts of a flush, and so you'll get some colonization. So there's a definite need for a new strategy, and Daniel has given us all the background and reason why. Excellent reasons why. And so one of the main things is to emphasize tree health. You need to keep your trees healthy. And in this case with lychee, at least in Florida, we're talking about periodic applications of potassium and minor elements because of our soil conditions make it more difficult, especially for the minor elements, to be taken up by the tree. The other thing is that nitrogen applications. Really, nitrogen applications should be reduced dramatically and in line with what are the nitrogen leaf levels. If your leaf levels are 1.1% or less, you might need, to apply a small amount of nitrogen. But I would suggest only doing it at the end of fruit set while you are controlling and preventing the mites from colonizing the panicles or if they're leafy panicles, right? There's also been some anecdotal evidence that applying iron during June or September may actually, in South Florida, I'm talking about, induce additional unwanted vegetative growth. So suggests that if you have noticed this as well, that you avoid iron applications during this period, because part of the strategy is going to be to have these trees synchronized with their vegetative growth and also to go dormant all at the same time. But if you're applying a nutrient that stimulates additional flushing, vegetative flushing, now you've got a problem in that you've got to protect this new flush perhaps, right? So we're also part of this strategy is, as we've mentioned, is you want to enhance the vegetative synchrony of the tree. In other words, you want all the stems to be at the same stage at the same time. You don't want a tree that has part of the stems growing, part of the stems stopped growing, part of the stems are flowering. That's not an efficient way or an economic way to manage the mite or the tree, right? So we're going to talk about synchronizing your tree. Also, irrigating mature trees. Really, you need to do this from flowering through harvest, but you really don't need to do it. I'm talking about mature trees, and I'm talking about South Florida after harvest, and certainly not during the fall and winter. You want to do everything possible to allow this tree to stop growing and become quiescent or dormant. Okay, so the idea is to limit sulfur applications to 2 periods: at panicle emergence through fruit set, and then the other time would be post-harvest, after post-harvest pruning, to synchronize all the vegetative growth of the trees after harvest. And Daniel spoke about this. You want to protect that new flush. And I know that when you harvest, you're pruning the stems. The thing of it is, though, you are not probably getting fruit off of every single stem. So the tree would not be totally synchronized. And what you want to do, we'll talk about next. So this is just some background on the optimum climatic scenario for lychee, right? So the idea would be that vegetative growth ceases after the summer vegetative flush, and the reason it slows down is due to cool temperatures. And of course, as I'm talking, dry conditions are also beneficial in stopping the tree from growing and becoming dormant. And then ideally, the trees would be exposed to 2 to 5 months of cool temperatures. And by cool temperatures, I mean temperatures below about 59, 60 degrees. And then as we warm up in the spring, there'd be moderately warm temperatures during the flowering period, which again goes along with what Daniel's findings has found. That's when the temperatures are in the 70s. So that's, a big problem for mite control, but that's what normally you want, warm temperatures to express the flowering. And then this would be followed by moderately high temperatures during the fruit development period. And below there is just a small chart showing some basic phenology of a lychee tree starting with June through March. So the FD is fruit development period. H is harvest. So you harvest in June basically here. And then June and July is when the tree is going to be flushing. So you need to protect that new flush. And then hopefully the tree is dormant from September through December. Sometime January, February, the tree begins to express flowering. And then March, April, May, and into June, you have the fruit development. A little more detail on this. So this is a similar type chart. So again, you have the harvest period in June, basically. And then, of course, the tree is going to flush during July, August, hopefully go dormant. During this period of August through December, the tree is building up carbohydrate reserves, which it needs in order to flower and successfully set fruit. And then you have hopefully cool temperatures during the winter, and you'll have then floral induction happening during that period. So that's, December, January sometime, and then panicle emergence happening sometime generally January, February, March, fruit set February, March, fruit development again, March, April, May. Okay. So shoot growth and monitoring. So as Daniel talked about, that you want to base your strategy on the conditions that are favorable for mite infestation and the tree's phenology. So what prompts lychee trees to grow? So stems to grow. So they'll grow in response to clipping the shoots to remove fruit, in response to pruning after the harvest, in response to over-fertilization, especially nitrogen, which needs to be severely limited if it's applied at all. Many years you may not need to. So I would suggest taking leaf samples to determine the leaf nitrogen levels. And they'll sometimes flush in response to a rainfall or an irrigation during warm and hot weather. So really the two major times that you need to be monitoring, of course, year-round is best, but monitoring the new growth at panicle emergence, and then after harvest and after the pruning. And of course, monitoring for the Lychee Erinose mite, as Daniel mentioned, you can see those symptoms on the leaves. You're not going to see the mites, but you can certainly look for the symptoms. So the recommendations for the Lychee Erinose mite control or this mitigation strategy is maintain healthy trees. You want to irrigate the trees from panicle emergence through harvest, because if you don't irrigate, you may reduce your fruit set, and you may reduce your fruit yields and fruit quality, right? So the trees, when they've got fruit on them and they're flowering, they need to be irrigated if we don't have rainfall, okay? So you want to stop irrigating after harvest, and basically don't start, and I'm talking about mature trees, start irrigating until next year's panicle emergence, okay? So I'll just make a caveat. If for some reason we enter a severe drought, something that's just unheard of, severe drought, you may need to irrigate slightly. But you want to try to irrigate not to the point where you're going to stimulate a new flush. Anyway, so you also want to be applying potassium and minor elements during the growing season, because those elements are used a lot by the tree. And so then you want to set up your tree for dormancy. So you've harvested your tree and you've clipped a lot of the shoots, but that's really not enough. That's not synchronizing the entire canopy. So you want to prune these trees after harvest to synchronize growth on all the shoots possible. Do not apply any nitrogen-containing fertilizer after harvest. Only apply nitrogen, as I mentioned before, if your leaf sampling shows that it's 1.1% or less. And again, I would suggest only during that, only applying the nitrogen, it would be during the fruit set or late spring, because the tree is going to be growing anyway, right? And I also mentioned the iron again, that in some cases we've seen after the summer and people have harvested the fruit, they've pruned the tree, and they apply iron, that it sometimes stimulates additional vegetative flushing. So this is just showing you a chart of what I just spoke about. And you'll also notice on there, you need to be looking out for the lychee webworm. I know this workshop's not about that, but that's something you should consider as well, because that can devastate producing fruit if you don't have stems that are expressing panicles. So anyway, so you'll see the lem control again. So we're talking about this late winter, early spring, January, February, March, and then post-pruning flushing after harvest. And then this pruning after harvest to synchronize the entire tree canopy. So I know I'm repeating myself, but I just want to emphasize that this would be the strategy. So you're only protecting the emerging, developing new vegetative growth with sulfur caused by the pruning, right? So after you have pruned the entire tree and you protect that response of new vegetative growth, then you would go ahead and stop. Then during winter, you want to monitor for the first signs of new shoots, panicle emergence. And you immediately begin sulfur sprays to protect the panicles, the flowers, and the set fruit. Then you can stop spraying the sulfur once the fruit is set. So that would be the 2 time periods rather than a 12-month time period of applying sulfur, or let's say some other compound once it's registered, but it would be during those 2 times. So here's a non-synchronized tree. You can see it has recently matured shoots and leaves. It has new shoots and leaves. it has matured shoots and leaves. It's not synchronized. That's a nightmare to try to control the Lychee Erinose mite. On the right-hand side, you'll see a grove where pruning was done after the harvest, and that's to synchronize the tree. It has other purposes as well, tree size control, things like that. But so far as with talking about the Lychee Erinose mite, has to do with synchronizing the entire canopy so that all the shoots are on the same page. This is just showing you on the left a dormant shoot terminal, and then on the right, some panicle emergence that's just starting. So you need to be out there looking at the shoots during especially the late winter to see when they're starting to express panicles. So here's this sulfur timing. So during the July, August flush after the pruning, and then during the panicle emergence. So this is a little bit of an aside. So this is a recommendation for some commercial growers if there is a need to disinfest a heavily infested tree. So you want to inspect the tree. You want to inspect for the symptoms of the Lychee Erinose mite. And this is the symptoms again. Daniel has shown these as well. And then you would, in some cases, if it's only parts of the tree, selectively remove infested trees and stems. And as Daniel mentioned, pruning without supplementary sulfur applications may actually aggravate the Lychee Erinose mite spread and intensity. So you want to remove that and destroy that pruned off material if you have a tree that's heavily infested or has large sections infested. And you want to disinfest your clothing and your equipment after, and then you immediately begin spraying sulfur of the infested tree, let's say, and I would suggest some of the adjacent trees around that infested tree. And you want to do that until the flush has completely hardened off. So this is the strategy. In this case, if you have a heavily infested tree, you want to disinfest. There's the tree in the center. That's the problem tree. And so then you would be applying sulfur basically to 9 trees. So procedures, the general management scenario for this heavily infested tree, monitor for the emerging shoots. At the new growth, begin the sulfur applications, continue applying until the tree has mature leaves. Acaricides registered for lychee trees. Daniel went over this. These are some other acaricides that are registered. They're not effective. They do not work. So far, the only one that's labeled that works is sulfur, although we're working with the IR4 program to hopefully get some of these other compounds registered. Again, caution with sulfur. Don't apply any oil compounds within 3 weeks before or after a sulfur spray because it will damage the leaves. And also during high temperatures, above 90 degrees, you can cause some phytotoxicity to the leaves. So you have to be careful about that. So for all commercial growers, you want to plan what you're going to be doing. You want to, in some cases, if it's just one tree or a few trees or just parts of a canopy, selectively prune out that material, remove that material and destroy it, apply the sulfur until that new flush is hardened off. So going forward, I think it's critical growers manage their lychee tree canopy to synchronize the flushing pattern. As Daniel mentioned, if you're just pruning or and not protecting the new flush that comes out in response, you're going to have a bigger problem with the Lychee Erinose mite. So I would suggest an annual pruning after the harvest. You want to reduce or eliminate nitrogen, especially near and after harvest. You want to reduce or eliminate irrigation, especially after harvest. So what we're trying to do is synchronize the tree and enhance that it's going to flush, and then go dormant. It's going to stop flushing, stop growing. And admittedly, in some cases, the tree, you're going to go ahead, you're going to prune, you're going to protect that new flush that comes out. And then it's going to mature. And then maybe 2, 3, 4 weeks later, maybe even 6 weeks later, it's going to start to flush again. I wouldn't apply sulfur on that new flush. I don't think that's going to be necessary. What you want to have is that one set of leaves that flush after the pruning, after harvest, that is healthy and doing really well. And don't worry so much about any subsequent flush. On excessively tall trees, large trees, let's say trees above about 18 feet, which are difficult to manage. They're difficult to spray, difficult to harvest. And potentially you're going to lose the lower part of their canopy. You could try to rejuvenate the tree and restart the lower canopy. If you're going to do that, you'll be out of production for at least 1 or 2 years. You want to whitewash the trunk and major limbs that you're going to cut down to, and then whitewash the tree. And then after you do that, then you would cut the tree, and then you would begin sulfur applications to protect that new shoots and flush coming out from that drastically reduced tree. And this is just showing you, we have an IFAS or Ask IFAS